All right. Dear members, colleagues, and good evening, good afternoon, good morning. Welcome to our ISDI virtual event, which is titled Methods of Analysis for Special Dietary Foods, How Science Ensures Food Safety and Quality for People with Special Dietary Needs. So today we are pleased to have participants from varying from the Codex family, the representative of Codex member countries, as well as colleagues from the Codex Observer organizations and also our ISDI members. So thank you all to take the time to join us today. My name is Jean-Christophe Kramer. I'm the Secretary General of ISDI, and it's a pleasure to be your host today. I'm also accompanied with our Communications Manager, Hugh Longton, who is helping us to ensure the webinar runs smoothly in terms of technical point of view. Just a few housekeeping remarks from the start. If you have any question for the speakers during the presentation, please click on the Q&A icon at the bottom of your screen, and you can type your questions. We will take questions during the webinar today. In case of technical issues, please use the chat and an ISDI staff member who will also respond to you privately. And finally, as you have probably noted, this session is recorded and will be made available after the event for the registered participants. So we'll share the link to the recording as well as the presentation and the relevant information with all participants. Before we start, just a quick recap of what is ISDI and what we do. Well, we are the International Special Dietary Foods Industries Federation, and we represent our national associations and of manufacturers of all types of what we call in Europe specialized nutrition, and in the international environment of Codex, which we would call special dietary foods. So you can see here all types of foods that we represent in terms of infant young child nutrition, foods for special medical purposes, dietetic nutrition, gluten-free foods, and sports food. We are also observer of Codex since 1973, and I often say that ISDI is itself a mini Codex with all the trying to navigate with the realities across the world in our field. So let's go to our topic of the day. I'm very pleased to have speakers with us today, and a high quality panel, I think. And of course, we organized this webinar also in the context of World Food Safety Day that took place on the 7th of June. Probably seen some postings around it, and so today that's why we are very pleased to welcome Sarah Cahill, the Secretary of the Codex Alimentarius Commission, who will give us some opening remarks. And then we will follow with the topic a bit more on the methods of analysis with Eric Konings and Dustin Starkey, who will introduce us on all the world of methods of analysis and what we do also within ISDI. After that, their interventions will hear from two standardization bodies, AOAC and IDF, how they are contributing also in the development of methods of analysis, with, of course, a particular focus on foods for special dietary users. And then we'll take questions through a panel. Eric will join us there. When Dustin at a certain time will take over again, and he will explain us our project that we wanted to launch today, or at least communicate here to all of you around a website, a public website, which is now in the final stages. And we will go live normally next week, but he will explain that in a moment as well. So let's start. I would like then to call Sarah and to share some opening remarks. And Sarah, we are all here. Thanks for joining us.
Thank you very much. So, dear participants, dear panelists, it really is a pleasure. It really is a pleasure to join you today to celebrate World Food Safety Day 2025. And although it may have just passed, let me nevertheless begin by wishing you all a very happy World Food Safety Day, because in reality, every day is about actions to keep food safe. This year's theme, Food Safety, Science and Action, is one that's very close to my heart, since having begun my studies for a bachelor's degree in science many years ago, science has influenced my view of the world and my work in many ways. And the mission of the Codex Alimentarius Commission has been defined as protecting consumer health and promoting fair practices in the food trade by setting international science-based food safety and quality standards. So science is at the core of standard setting. Science is at the core of the Codex Alimentarius Commission and its work. And I would say science is at the core of food safety and food quality, including nutritional quality. In fact, I think it's fair to describe many of the standards, guidelines, and codes of practice of the Codex Alimentarius as an example of science in action. Without science, without the scientific advice provided by a whole range of FAO and WHO expert bodies, without the data collection and research efforts of members, academia, and the food industry, which includes many of you here, we would not have over 14,000 quantitative codex standards, including MLs for contaminants in food and maximum limits for additives. But of course, standards are only as good as their implementation. And methods of analysis, the focus of the webinar today, play a critical role in that regard. Methods of analysis enable us to verify compliance with standards, so helping ensure products meet the food safety and quality requirements, including those for nutritional quality, as well as authenticity markers. Without such methods, we would have no idea whether a commodity was meeting provisions that had been established to ensure the food was safe or meet the quality parameters or nutritional requirements. And when it comes to consensus building and avoiding trade problems, it's not only important that we agree on the safety and quality requirements, but that we also agree on the way in which we determine that those requirements have been met. Because not any method will do. We need methods that have been agreed internationally and have been reviewed to ensure that they are fit for the intended purpose. And having these internationally agreed methods contributes to harmonization and the facilitation of food trade. Now, methods, of course, can fall into a number of categories. We have defining methods, as we call them in Codex, which is the only method for establishing an accepted value of an item to be measured, or a reference method, which is the recommended method where there is maybe no unique defining method. And these methods are very important in cases of disputes, for example, or also for calibration purposes. However, they may not be optimal for routine day-to-day use. So within Codex, we also define methods that may be used for control, inspection, or regulatory purposes. And this enables other factors to be taken into consideration when recommending methods, such as accessibility of methods and also the needs of low and middle income countries. Within Codex, the establishment of numeric performance criteria for methods enables more choice of methods while still promoting harmonization. But Codex does not develop methods itself. Rather, it relies on the methods developed by standards development or methods development organizations, several of which we have represented at this webinar today. And in line with our procedures in Codex, the members endorse internationally validated methods of analysis within the different categories, such as a defining or a reference method, or a method for routine use or control purposes. And in order to do this, cooperation with other organizations is key. Organizations responsible for developing standardized methods have been at the heart of the work of the Codex Committee on Methods of Analysis and Sampling for many years. Within this committee, CCMAS, efforts are ongoing to improve its processes to ensure consistency in the ways of working, namely in its assessment and endorsement of methods, which we believe can lead to greater reliability of the recommended Codex methods. Within Codex, the aim is to recommend methods for analysis for which the reliability has been established with respect to a number of criteria, such as selectivity, accuracy, precision, limit of detection, sensitivity, and practicability and applicability under normal lab conditions. And applying these in a rigorous manner ensures that members can be confident that the recommended methods of analysis are truly fit for purpose. Within Codex, we're currently working towards establishing a single reference for methods of analysis and sampling, and an eventual database of these for ease of use. We are, however, finding this is not maybe as easy as it seems. And I think this highlights the importance of ensuring consistent use of terminology, among other things, as we work on methods. So this work will continue in the near future. What observers such as ISDI are doing with this webinar, and it's soon to be available one-stop shop for methods of analysis for foods for special dietary purposes, is very complementary to Codex efforts and well appreciated. It in fact contributes to goal 3 of the Codex strategic plan of building awareness of and promoting the use of Codex standards. And such initiatives which help Codex recommendations get to the intended user or make them closer and more accessible to the user are to be applauded. And we really value this cooperation and encourage the continued collaboration in this regard. But nothing stands still, and this is more true than ever. We need resources that can be easily updated in pace with the latest developments. And even within Codex right now, for example, a review of methods of analysis for nutrition standards that have been developed by the Codex Committee on Nutrition and Foods for Special Dietary Purposes is currently underway, which may lead to some changes in recommended methods. Hence, ongoing collaboration is essential to ensuring that we keep pace with development in the areas of methods of analysis and ensuring we share that with all our stakeholders in a timely manner. So I look forward to the panel discussion as it takes a more deep dive into the important role of methods of analysis as well as their timely accessibility. Once again, happy World Food Safety Day and wishing you a successful and informative webinar. Thank you.
Thank you, Sarah. Thank you for your kind words and introductory remarks. It was interesting to see that, of course, during Science in Action, of course, the World Food Safety Days team of this year, we also look at the implementation of standards and the fact that methods of analysis are there, as you rightfully pointed out, essential to implement them, particularly in our field, of course, for special dietary food. So we'll come to that in a moment, but and of course, as an observer organization, we fully support the Codex ambitions and strategic goals, and that's why we also indeed are happy to organize this webinar to interact with the Codex members, and happy to have you here also in support of us. Thanks again, Sarah. It is then my pleasure to start the content with with Eric Konings, who is now an independent consultant but has a very long experience as an expert at the Nestlé Institute of Food Safety and Analytical Science, and he will talk us more about how science-based methods of analysis support the food safety for our targeted population. Eric, the floor is yours.
Thank you, Jean-Christophe. Good day, everybody online. Happy to be with you today and elaborate in the next few minutes how science-based methods of analysis support food safety and quality for consumers with specific dietary needs, and particularly, of course, the products they use. We heard Sarah about Codex standards, which are mostly the basis for national regulations. They include safety and quality requirements for a food product. For example, minimum and/or maximum levels of a nutrient in infant formulas. Codex committee standards shall include a method of analysis, which is listed in the Codex Standard 234 to check compliance with the relevant provision in a standard. Results of methods of analysis must be reliable to ensure safety, quality, and fair trade. We heard Sarah saying Codex Committee of Methods of Analysis identifies appropriate methods of analysis to do that. How do analytical methods support safety and quality of products serving specific dietary needs? Please go to the next slide. Next slide, please. Yep. For that, it is important to realize that in the production processes of a product, three main sources contribute to the variability: materials, and analytical methods, and the process itself. They contribute to the variability sources in the production. Understanding these sources helps in controlling and minimizing variation, leading to more consistent product quality and increased efficiency. Next slide, please. You see on the slide a Gaussian curve indicating analytical variability, reflecting the distribution of data produced by an analytical method around a central mean, which is the true value exactly in the middle of regulatory minimum and maximum. The width of the curve is defined by the standard deviation of results produced by the analytical method. Values close to the mean are more frequently found than those far from the mean. Usually 95% of the analytical results produced by the analytical method fall within 2 standard deviations from the mean. You can imagine if the variability or the standard deviation of results by an analytical method goes beyond the process variability or even the regulatory limits, you can find an analytical result outside the limits, even though the true value is exactly in the middle of the regulatory requirements. This can result in misinterpretations of the quality of the product and may lead to incorrect judgment of non-compliance with regulations during enforcement, leading to various consequences, including financial penalties, legal actions, reputational damage, and even market access problems. Therefore, we need accurate and precise methods. And with this, I would like to hand over to Dustin.
Thank you, Eric. And so Dustin will talk a bit more about the modernization of analytical methods for FSDU and through the stakeholder program on infant formula and adult nutrition, so SPIFAN. Dustin, feel free to start. Before we start, if members or participants have questions, you can always use our Q&A and we'll make sure we can address them a bit later. Dustin, you have the floor.
Thank you, Jean-Christophe. And hello, everyone. Hugh, you can go on to the next slide. So you just heard from Eric about some of the challenges of test method variability and meeting nutrient specification limits. I'll now talk about the work by the special foods industry and international standards organizations to reconcile some of these challenges. The Stakeholder Program on Infant Formula and Adult Nutritionals, or SPIFAN for short, was launched by AOAC International in 2009 to modernize test methods for nutrients in foods for special dietary uses, including products like infant formula, follow-up formula, and foods for special medical purposes. The demand for SPIFAN was driven primarily by the infant formula industry, and this was because the complexity of formulas had evolved beyond the capabilities of existing test methods, making them no longer fit for purpose in some cases. Additionally, regulatory challenges between infant formula companies and country authorities suggested a need for harmonized test methods. And so SPIFAN set a course to validate new test methods through a scientifically rigorous consensus standards process. This included the establishment of a tripartite agreement between AOAC, ISO, and the International Dairy Federation to jointly develop and approve methods. The program also convened global subject matter experts from industry, academia, and regulatory agencies to do the hands-on work. So all these factors, a rigorous validation process, 3 internationally recognized standards organizations, and global subject matter experts from across industry sectors has helped ensure the integrity, credibility, and acceptance of the methods, including by the Codex Alimentarius. Next slide, Hugh. This slide shows a summary of SPIFAN methods that were published as official methods by AOAC, ISO, and IDF since 2009. In total, there are more than 25 official methods, and 21 were adopted by Codex as Type 2 or Type 3 methods for infant formula. And all of this demonstrates the overall success and credibility of the program. These methods were authored by scientists from roughly 10 different organizations and validated using many different liquid and powdered infant and adult nutritional products. You will notice that almost all the analytes show more than one method number, and this is because each SPIFAN method was published as equivalent standards by AOAC, ISO, and IDF. Those highlighted in bold font were adopted by Codex for infant formula, and of those, all but 2 were adopted as type 2 methods, which means that they're recommended for use in cases of trade dispute. And then 2 of those methods were adopted as type 3, which are also recommended for trade purposes. The Codex adoption of SPIFAN methods is important because it helps promote global harmonization of the very best test methods for testing infant formula. And this is important for lowering trade barriers as well as ensuring product quality for a sensitive consumer population. More recently, nearly all the SPIFAN methods adopted by Codex for infant formula were endorsed for follow-up formula by the Codex Committee for Nutrition and Foods for Special Dietary Uses, as well as the Committee on Methods of Analysis and Sample. And the final step in the process will be approval by the Codex Alimentarius Commission later this year. Next slide, Hugh. The success of SPIFAN continues today with the ongoing development of methods for priority nutrients like the amino acid taurine, plus novel nutrients like human milk oligosaccharides and phospholipids. There's also several methods being developed for contaminants like chlorate and MCPD. The goal of this work is to publish the methods as official standards, just like those methods that I summarized for you on the previous slide. Also, the SPIFAN community has recently discussed an evergreen process to revisit and refresh standards that were published in the early stages of the program. I'd also like to note that the collaboration between AOAC, ISO, and IDF that started so many years ago continues today. So in summary, SPIFAN is still going strong and will continue its work and successful track record. I'll now turn the program back to Jean-Christophe to introduce the panel. Thank you.
Thank you very much, Dustin. Thank you very much. So here we have received a bit of a snapshot of the relevance of methods, why they are important, and by Eric and also Dustin explained us how we organize this, the industry and the standardization bodies, and focusing on this program which we call SPIFAN, and some of those in CCNFSDU and probably also CCMAS have already heard about this acronym. Now, as we said, we work with the standardization body, so we are very happy to have with us Katerina Maslovska from AOAC, Deputy Director, and Aurélie Dubois, the Science and Standards Program Manager at IDF. Before we go into real questions, we wanted to give the floor to the 2 ladies, Katerina and Aurélie, to introduce, what exactly they do, what is their role in the program, and how their entities are contributing in the development of the standard. Katerina, maybe we can start with you, and I'll give you the slide here that you can introduce to the audience.
Thank you, Jean-Christophe. This figure shows AOAC's multi-step process through which SPIFAN and many other AOAC official methods are developed and validated. The process begins with inputs from stakeholders on which method needs to be developed, defining the method scope in terms of analytes and matrices, identifying regulatory requirements and other important considerations, which are then used by AOAC technical working groups to develop method requirements, which are provided in a document which we call Standard Method Performance Requirements, or SMPRs for short. SMPRs are AOAC's voluntary consensus standards that provide minimum requirements that a given method should meet. SMPRs are used as criteria by method developers to develop and validate methods, and also by AOAC's expert review panels to evaluate candidate methods that are being considered for AOAC official method status. Methods and corresponding validation data are submitted through open calls. The initial submission and expert review panel evaluation are typically based on a single lab validation study and can lead to a method being approved as first action AOAC official method. For final action status, we need reproducibility information. So a collaborative study involving multi-lab validation needs to be conducted for the target matrices to evaluate reproducibility and to generate reproducibility data, which are again evaluated by the expert review panel that can recommend the method for final action. The actual decision on final action status is rendered by the official methods board, which does the final review and approval. So as you can see, the process is quite involved. And if we are developing a method jointly with ISO and IDF, we collaborate with these organizations throughout the entire process by sharing expertise and also aligning at important process steps, such as before the collaborative study. And if applicable, we also collaborate on a joint submission to Codex.
Thank you, Katerina, to explain us a bit more in detail how you're working in AOAC in a very structured and in a very straightforward way. But that helps us to understand a bit the development of it. And now I'll give the floor to Aurélie to introduce about more how you work in IDF on the development of the methods. Aurélie.
Thank you, Jean-Christophe, and hello everyone. And thank you, Izzy, for the opportunity to be here today. First, I just want to say a few words about IDF and to explain why I'm here on the panel today. IDF is a non-profit international non-governmental organization that represents the global dairy sector since 1903. We do have a network of about 1,200 experts from 40 member countries who represent themselves 2/3 of the world milk production. So of course, milk is a key ingredient or component of infant formula and follow-up formula, for instance. Our expertise spans through the full dairy chain, involves farmers, researchers, manufacturers, processors, governments, and more. Because the mission of the organization is really to be the voice of the dairy sector, in particular to intergovernmental organizations such as FAO, WHO, and of course Codex, with standards being at the heart of our activities. We have a very strong and historic link with Codex, with the development of many dairy-specific standards, but also continuous involvement in many other committees relevant for the dairy sector. We also have our joint program with ISO to develop joint standard. That's the same document in 2 numbers that you have seen in the table presented by Dustin. So standards for methods of analysis and sampling for milk and milk products. And many of which, as Kate explained, are developed in a collaboration with the OSC and other organizations. We also collaborate with the World Animal Health Organization for animal health standards, a slightly different area. But international standards do play a vital role in safeguarding our food and providing trusted framework to produce safe food and to strengthen consumer confidence, as was said before, especially when the consumer is an infant or a young kid. Our organizations, AOSC, ISO, ISD, IDF, collaborate to ensure that all relevant stakeholders are involved. Analytical experts, manufacturer, ingredient suppliers, equipment and test suppliers, authorities, etc. Actually, Codex did make this request to us back in 1963 to ensure we provide harmonized methods to facilitate standardization. IDF and ISO are organized slightly differently than AOSC. Our members are our member countries. We have different processes, but through our collaboration, we have managed to identify key steps to align our procedures and align to ensure that we have the right objectives aligned. For instance, AOSC has this unique process to identify the standard method performance requirement. So we ensure that we identify the right experts to contribute, that from the beginning, all the aspects are covered, that we don't oversee a regulatory aspect or a different aspect that could have been missed. And then once a protocol is identified through the call for method, We ensure that we have a consensus. So it's reviewed by AOAC membership, by ISO membership, by IDF membership, really covering a span of on geographical aspects, on expertise, and stakeholders in general, really to make sure the work is relevant. And this is really a key step, because once we have an agreed protocol, that's the one that will be going to the multi-laboratory study, the validation that Kate explained, to ensure we have a robust method that can be used throughout the world by different laboratories, different regions, and provide this comparable and reliable results. And that's really a key unique language if you want to compare apples to apples and ensure that you have a reliable result, especially for safety aspects or nutritional requirements. So that's really the key review. And then when we do this validation, we ensure we have the right samples identified, and that the laboratories really reflect that diversity globally. And then we have a final step after that, once the result of the study through the statistical treatment, I won't go into those details today, that really the standard is approved by everyone and pre for publication. And as Kate mentioned, the last point is when it's relevant to a Codex standard. So if it's a method that allows to verify the compliance of a provision of a Codex standard, we also collaborate to make sure we have a clear submission, a clear explanation why this method needs to be into the Codex system, because it is more precise, because it will provide a better result and allow a safe product traded internationally. So without this, my final word would be that all stakeholders really involved in standardization community contribute to food safety. And through all of these organization, you can contribute, you can be an expert, you can drive the work, you can bring your stone to these and help make food safer. Thank you.
Thank you, Aurélie. Thank you for this good also concluding remarks, and of course explaining how IDF works and the whole approach on how is your perspective here in the dairy sector. And as you know, dairy ingredients are always an important ingredient in our, particularly of course in the infant formula. So let's go back to our panel. We have well, good in time to get a few questions. I'll look so we can spotlight everyone here on the on the screen. So I have a few questions to our panelists. I'll feel free to reply to the best you can, and you can also complement each other if you believe so. The floors and the microphones are open. My question maybe to Katerina firstly. What is your because you explained very well the process of how AOC develops a standard. What is your time frame in developing such methods throughout the process? So is that always straightforward, or how much, or are there many discussions in between? Can it take years?
Thank you, Jean-Christophe, for the question. Yeah, it's a very good question. The time frame obviously depends on many different factors. For example, I described that we developed the SMPR at the beginning, and so it depends on how complicated the development is. Sometimes it takes, let's say, a few months. Sometimes it can be up to a year. So it depends how complicated the discussions are. Then also, are there any methods available to meet the requirements, or do they need to be developed from scratch? Right. So that also will affect the timeline. And then how many times does the expert review panel need to meet to actually evaluate the method, have a discussion, provide feedback back to the developers? They may need to go back to the lab and generate more data. So it also depends on these factors. And then obviously one of the biggest, I would say, factors on the timeline is the multi-lab study. How long does it take to prepare it? How long does it take to actually conduct it? And then also evaluate the data, prepare the manuscript, and so on. But in general, if everything goes smoothly, the process from the start of the SMPR development to the final action method, I would say can take around 3 years. And I don't know if someone else will have a different number, but I would say around 3 years is a realistic time frame for development of a method from the beginning to final action. I don't know, Aurelie, if you have a different perspective from your side.
I would actually agree with this timeline and agree with that there are several factors. Another key factor is the availability of the we call them action team leader in IDF ISO, or the project leader, if you wish, the champion, because that will really impact a lot the process and also the participation of experts and the comments received. But I think 3 years is a good number. Sometimes it could be shorter, sometimes a bit longer.
Eric, from your experience.
No, yes, that's certainly. I would add another 2 years, maybe until Codex adoption. So if everything well is planned, so we have to think about 5 years for the whole process in general.
True that we always aim to seek, call it the cherry on the cake. The cherry on the cake is getting a Codex. A reference in the Codex standard 234, isn't it?
Yeah, yeah.
Good. Maybe another question, probably for Eric. How quickly does the science behind the methods improve? Because we always science in action is the theme of this year, so there's always action. Do you see that from your experience? What is then the typical timeline of a method, or the lifespan of a method, and is it improved regularly?
Yeah. Thank you, Jean-Christophe. Yes, science improves relatively quickly, but if we discuss methods, in this case, standards for testing food source special dietary uses, I think the AOC SPIFAN project started in 2010, and that was a follow-up. Let's say, originally at that time we concluded earlier standards, they originated from the 1980s, 1990s. One of the examples I can say perhaps is around B vitamins. Around that time, 1980s, you had microbiological test methods for B vitamins in these kind of products. And you had a method for each of the B vitamins. And also, compared to the methods which came available, let's say 2 decades later, they were time consuming and the precision was not too good. So 2 decades later, we had technology as HPLC, and now we have a standard where vitamin B1, B2, B3, and B6 can be done in one single run, which is cost efficient, time efficient, and they have a better precision. So I would say, and certainly when you think about 5 years to develop a new standard, so this showed, and in the 1980s, these methods were developed when in the US, the new Infant Formula Act was enabled. So it took, let's say, 20, 25 years to start new standard development based on new technology. So I would take it in that frame. I hope this explains a bit how quickly these standards move as well. Of course, based on regulatory requirements, if you need suddenly, let's say, other technologies to ensure and demonstrate compliance, it can be quicker, of course, and you can decide to change or update the standard as appropriate.
Thank you, Eric. Maybe a question to Aurélie. When we look at the development processes, how do you think, how would you compare IDF towards AOAC if we take that as 2 examples here for 2 standardization bodies? Could you elaborate a bit more, maybe?
Sure. So I think we have an overall similar process, consensus-based, with experts involved and several key steps of review that we have briefly explained. I think the key differences are from the AOC perspective. They are establishing the set of criteria, the framework that will describe what do we need a method for. This is something that we don't really have in IDF ISO. So they set the set framework and make the link with regulation or codex, for instance. So I think it's a very good process that allows for the call for methods. In IDF ISO, we are taking proposals and then check if it fits with the codex or check the regulation, or we also sometimes do some gap analysis. But I think it's a very good process. So we are happy to tag along on when we have a shared scope. Another maybe difference is that we have in IDF ISO, IDF procedures are completely aligned with ISO, since we are both member-based organization, not companies. So our members are consulted and then they consult the members. So it's a sort of a double consensus based. We consult experts internationally, and each of our member countries also consult their own base. So it means there's a little bit more time involved in one or two steps in the ISO process. So it takes a little bit more time, but we should not forget it also reach more people, more stakeholders, and also facilitate acceptance of the method. I think that's the two main differences that come to my mind. I don't know if Kate has any other.
Yeah, I would just add that, that's why it really is important to align and work together, right? So we actually align in the right moments in the processes, because as earlier said, we started the SMPR development. At the time, we already basically engaged, IDF and ISO, right, to get experts into the groups. I think what's also important is that many of our experts are actually shared experts, working basically within each organization. So I think that helps as well in alignment. And as already mentioned, having a really good champion for that given method, in each organization, and it's actually one person, right, or a group of people. It's hugely important because those are the drivers, who can take the projects through.
Just looking at the time, we can maybe one small question. Well, small. It's maybe a difficult one, but maybe for Eric, how are methods also leading to innovation in the development? So.
I think for that, it could be, if you think about new ingredients, for example, you see, for example, HMO, the human milk oligosaccharide, is the third most compound in breast milk. So it is a complex matrix. So method of analysis help to find out the structure, to analysis, let's say, have more clear how they can be applied in, for example, infant formula. So there is a lot happening on that, where method of analysis supports, let's say, the addition of potential new ingredients in these kind of products to make them more, let's say, comparable to breast milk.
Interesting. Thanks, Eric. I'd like to give the floor now back to Dustin, because he's ready to explain us our project here in ISDI, what we have done over the last couple of months, which was actually to develop a public website. But Dustin, you will explain much more, and you will share the slides in a moment as well.
Yes, thank you, Jean-Christophe. It's my pleasure to introduce you to our new test methods website. This site was developed in collaboration with our experts and the support of AOAC, ISO, and IDF. We developed the site to meet a very clear need, and that is to help anyone working with foods for special dietary uses by making it easier to find reliable, validated, state-of-the-art analytical methods. And this is important because using methods that are not fit for purpose can have serious consequences, like erroneous product recalls that damage brands, incur financial loss, and erode public trust, The site is designed to mitigate some of those risks by helping users find the right expert recommended test methods for testing FSDU products. So in short, it's a one-stop shop where regulators, labs, manufacturers, and others can quickly find and access trusted, validated methods. And this supports a higher level goal to globally harmonize labs to the very best test methods for FSDU products, ensuring greater consistency, reliability, and quality. Currently, the site scope is limited to methods for infant and follow-up formulas, but our goal is to add methods for other FSDU product categories by the end of the year. And we also plan to expand the scope beyond nutrient methods by including contaminant methods in a later phase of the project. The site is designed to provide a list of methods and a brief description of each. It's important to note that the site does not actually host the methods themselves, but rather provides links to them on the standard organization's websites. And this helps ensure the end user is always accessing the most updated method versions. The site is also designed for simplicity and with easy search and browsing functions so the user can get to the information they need quickly. So we'll now take you on a brief tour of the site so you can see how it works. And we'll start from the home page and show you how to do a search and then walk you through what a typical results page looks like. And we'll also introduce you to some of the other features of the website. Hugh, I'm not seeing the, are you sharing?
Yeah, we seem, oh yeah, now we start, yeah, okay.
Excellent. Yes, perfect. So everything you need to navigate the site is on the home page. You have the option to browse by commodity, analyte, or test method, which you can do by clicking on one of these three buttons or alternatively from the menu bar. Using the analytes as an example, you can utilize a search bar at the top to narrow it down or have the option to browse alphabetically. So say you're interested in the nutrient biotin, we could type it in the search bar, or we could just click on the letter B to search alphabetically. Here it will show you that for biotin, there are methods relating to both infant and follow-up formulas. And if you click on biotin, it will then list all the applicable methods. Equivalent methods will appear on the same line item. There's also a link to access the methods directly from the listing. But before we look at the method description page, we're actually going to take you back to the home page to show you other ways to find methods. Again, you can search directly in the search bar or use the filters to narrow it down. And when a commodity is selected, only the analytes available for that commodity will be shown. We can also select the method principle here. And the principle refers to the primary analytical technique or instrument prescribed by a test method. Selecting infant formula and then biotin will bring up the applicable methods listed below the search. Going to the final results now, it provides all the essential information about the method, including how it relates to Codex Alimentarius, as well as a link to access the method. You can also contact us if you have any questions about the method by clicking on Contact Us at the bottom of the page. You can also export or save the results as a PDF by using the Export to PDF button at the top. And in addition to methods, there are more pages under the Info tab in the header. This includes key terms and definitions, some frequently asked questions, and information about the site and how to use it. Finally, we will post updates in a blog section from time to time related to the site and methods. We also plan to add French and Spanish translations later. If you'd like to be kept informed, then please subscribe to updates and you'll be alerted when we add new commodities, contaminants, methods, and other updates. You can also subscribe from the footer on each page. So with that, we have come to the end of the demonstration, and I will now turn the program back to Jean-Christophe for questions and answers and conclusion of the webinar. Thanks very much.
Thank you, Dustin. Thank you for yes, my video wasn't on. So thanks, Dustin, for your for explaining the big project we had over the last couple of months, which was to list all the available methods. It seems very easy, but actually was there's always a bit of preparation before that, and making sure that all the methods are you know, correctly listed with the accurate number. It's still work in progress. We aim to go publicly next week when the first part of this project is being finalized. We're focused on infant formula. The other foods for special dietary users will follow. So it's but we need to make sure that we triple check all the things before they go public. Of course, there's a but we think that this website will help not only, of course, industry, but also the regulators and the participants here at this webinar to understand a bit more which method is used when and for what, with purpose, which recognition, etc. So this is really our ambition here. So feel free to reach out to through the website to us, to the Secretariat here of ISTI. We'll be answering, of course, whatever we can. And we have our experts with us to support us in all the queries you may have. We're almost at the end. I don't know if, well, we've seen a question on the Q&A from one attendee, which is more on the implementing phase. I'll read it out. What happens with the issue of getting false results for a batch due to analytical variability by preventing if we take multiple samples per batch and take the average of these results, so if so, how many samples per batch would we recommend when assessing compliance of infant formula to regulatory requirements? I suppose this is more of a question of how the implementation is and the discussions we have. So here with SPIFAN, we develop them, but of course, when we apply them, we need to make sure that we have also a consistent approach on that. I believe Eric could probably provide an answer.
Thank you, Jean-Christophe. So I don't have an answer in how many samples. I think also depends on the variability, the standard deviation of a method, but also how this compares to the regulatory requirements. So in the case you have a very tight regulatory requirement and a huge variability, I think we have seen examples that it is up to a chance of 20% that there will be a result out of the regulatory, let's say, requirement. So this will, I'm not sure if this can be solved by multiple sampling. I've not been calculating this, but worthwhile to look into this. Thank you.
Thank you, Eric. I don't have many more questions at this stage, and we are coming to the end of the webinar. So my proposal is to conclude here with sharing, of course, post-World Food Safety Day. We want to congratulate everyone about all the job they do in making sure that the people of use of special dietary foods can of course, continue to get an access to the foods we manufacture. I think it's essential for us that and it's our responsibility and our duty as an industry to provide you the best safe foods appropriate, especially for those in need, being the most vulnerable, but also the patients or even our sports people that require appropriate foods to perform. Methods of analysis, as we have heard, are really relevant and important. Sometimes very technical, difficult to understand, but it is essential to make sure that we are able to comply with the requirements in the Codex standards, but also at regulatory level in regional or national jurisdictional systems. But with that, I really want to thank all our panelists and our hosts. I mean, Sarah, thank you for taking the time to be with us. I know you have a very busy schedule. So thanks again for all your kind opening remarks, very accurate as always. And also I wanted to thank Eric, Dustin, to give us a bit of a more opening the perspective how we work within this industry to ignite and steer the development of methods. And then also with Kate from AOAC and Aurélie from IDF in helping also to get the standardization bodies with us in order to make sure that we have relevant, appropriate methods. And then, of course, hopefully to get them endorsed by Codex as well. So thanks again, everyone, for your participation, the questions. And I'm sure we'll meet very soon, if not a bit later in one or another Codex meeting, be it CCNFSU or CCMAS. Thanks again, and I wish you a very good rest of the day. Thanks again.
Thank you. Bye-bye.
Thank you. Bye.
Thank you. Bye-bye. Bye.